I presume about 1,000,000 times that you've heard of chronic lymphocytic leukemia, and generally everyone knows that it's a type of cancer. There are both people who are in the clinical field and others who are not but have interest in knowing about the health-related implications on chronic lymphocytic leukemia. We will spend these few minutes to have a short discussion about chronic lymphocytic leukemia. As the name suggests, it is chronic because the cells that are involved in this type of leukemia are matured cells. They take over years to develop. And it is lymphocytic, self-explanatory, because the cells here are lymphocytes. And the reason why they are leukemic is because the cells are uncontrolled proliferating, producing majority of these type of cells and overcrowding the bone marrow. In discussion of chronic lymphocytic leukemia, to make it so easier, there are two major processes that you have to understand, just to have an idea of it, and these are the two major processes that relate to its clinical implications and prognostic factors. We will start by the haematopoietic process in the lineage of lymphocytes. That is the first process. The second one will be the genetic aberrations or alterations that really affect the cause of chronic lymphocytic leukemia. In discussing lymphocyte production, let's begin with this. Lymphocytes are produced in one major organ, the bone marrow. This is where all lymphocytes are produced. We have two types of lymphocytes. We have the B cell and the T cells. Both cells develop from the stem cell of the bone marrow, and through maturation, the B cells remain in the bone marrow and mature, while the T cells migrate to thymus and their maturation occur within the thymus. That's why T cells are also called thymocytes. After their maturation, there is another step we call differentiation. Don't be too confused. Maturation and differentiation are not the same, but as I discuss along, you get why we call one maturation and the other one differentiation. The cells begin its development within the bone marrow from blastic stage. When all factors are favorable, they develop into mature cells. And these mature cells, they will have to travel to another set of organs that we call the secondary lymphoid organs. The secondary lymphoid organs are three. We've got the lymph nodes, the spleen, and the tonsils. Why do we classify them as secondary lymphoid organs, and what is special about them? The secondary lymphoid organs have got a microstructure in them that we name as germinal center. This is where actual differentiation occurs. In the germinal center, which is the microstructure within these organs, there is an interplay of changes of these mature lymphocytes that would at the end classify them into two main cells. This is how we obtain plasma cells, and also we obtain the memory B cells. Let's talk through what processes occur in there. In the germinal center, we've got one, the T follicular lymphocytes, CD4. They produce cytokines to help in this differentiation. We've got dendritic cells. They present antigens to them that help them develop actual immune response to these type of antigens that are presented to them. Stromal cells regulate the cell movement in this microstructure that we call the germinal center. The two processes that will occur within the germinal center are somatic hypermutation and affinity-based selection. Through the germinal center process of differentiation, these lymphocytes at the end obtains a BCR, that's the B cell receptor. This is where the somatic hypermutations has occurred in several stages, and therefore different types of lymphocytes have been obtained, and they are able to have affinity for different antigens that are presented as they migrate back into our system, and they are ready for immunity or immune response. Now, we have discussed how these lymphocytes under normal circumstances do mature and do differentiate and are now exact at potency to elicit their immune response. Any lymphocyte at the germinal center which does not undergo this somatic hypermutation and affinity-based selection will now be channeled through a programmed cell death that we name as apoptosis. So this is one of the well-defined cell death in our body to get rid of abnormal cells. Note this because this will make sense when we get to chronic lymphocytic leukemia explanations. Now we are back into the second aspect, that's the genetics. And I believe you're going to love it because it gives more light to chronic lymphocytic leukemia. But this is what you should know. In the human cells, we have organelle that we call the nucleus. Inside the nucleus is a crystallized structure we call chromosome. Within the chromosome, we have the DNA, and a portion of the DNA contains instruction, and this portion of it is called the gene. The gene has nucleotide base pair, and this nucleotide base has instructions regarding protein synthesis, and lymphocytes are obtained, or lymphocytes are produced by these instructions, and they are regulated in their maturations by these instructions. But what happens within this that affects chronic lymphocytic leukemia? There is a chromosomal aberration called deletion. The deletion takes away a portion of a chromosome that contained this instruction. What are the chromosomes that are involved in chronic lymphocytic leukemia? We have three main. We have chromosome 11, chromosome 13, and chromosome 17. In chromosome 11, there is a gene called ATM, ataxia telangiectasia. This ATM gene ensures that broken double strands of DNA in these lymphocytes during their maturation are repaired, and therefore if this portion of the chromosome is deleted, it means there will not be any DNA repair, supposing there is an error during maturation of lymphocytes. Therefore, broken pieces of the DNA will still remain in these lymphocytes as they mature. One abnormal lymphocyte obtained in this process. The second gene we will talk about is chromosome 13. Chromosome 13 contains microRNAs. MicroRNAs inhibit BCL proteins. BCL gene produce BCL proteins, and BCL proteins are anti-apoptotic proteins. Anti-apoptotic means it doesn't allow the programmed cell death that is established to get rid of cells that become abnormal in the process of maturation. Meaning, if there is no inhibitor like the microRNA from chromosome 13, the BCL anti-apoptotic protein is now a champion, inhibiting abnormal cells from undergoing cell destruction, and they remain and they mature even though they are abnormal. Now, the last chromosome we are going to talk about is chromosome 17. Chromosome 17 codes for the production of a TP53, the TP53, the tumor protein 53. This is also called the guardian genome. The reason it is having this appellation is because it triggers the programmed cell death. It is the key that switches the apoptotic process to begin so that abnormal cells are removed. But on chromosome 17, the section where this gene is found undergoes a deletion, meaning it is getting rid of this guardian genome. Therefore, nothing is going to switch the apoptotic process to begin, and therefore abnormal cells thrive and overgrow within the human body. Now we can make a sense out of it, that we understand the cellular process from the stem cell how lymphocytes are generated, and the genetic aberrations that do affect this cause of leukemia, why abnormal cells thrive. But the question not answered is, how does this deletion occur? Nobody knows. And therefore, we cannot really associate CLL to a lifestyle or a behavior or a conduct that has been the cause of this. Probably underdiagnosed in some places and had been probably associated to a different cause, but the symptoms are experienced. The noticeable symptoms of this disease is as a result of the overcrowded abnormal cells within the bone marrow, within the thymus, and within the secondary lymphoid organs I mentioned as tonsil, lymph nodes, and the spleen. Now, if the bone marrow is overcrowded by massive abnormal lymphocytes, a large space is occupied and not enough space is left in the marrow for production of red cells. The patient will be anemic and they will feel weak. That is why we say tired all the time. At the same time, there is no place for production of thrombocytes, which are platelets. At long run, the patient will be at risk of bruises or bleeding, and this is also a symptom of that. Overproduction of these abnormal lymphocytes will also travel into the secondary lymphoid organs or tissues. They accumulate within our lymph nodes, and we have cervical lymph nodes which will swell up as more cells accumulate. Your stomach can have a distension because of enlarged spleen, and even a man can look pregnant, and a female who is not pregnant can appear as though they are pregnant. And as well, your tonsils can also have accumulated cells, which is not really common, I will put it that way. Now, what are the laboratory findings in this? We've already mentioned it: low hemoglobin, low platelets, high level of lymphocytes, and on examining a blood film, you will see a surge of lymphocytes. Some of them will look well matured and high nucleus to cytoplasmic ratio, meaning almost all the cell size has been occupied by a nucleus and the nucleus look clumpy. In other ways, you can also find a film of CLL where the cells look quite atypical. They can have larger size, prominent nucleolus, but we interpret not in separation from other findings, and also genetic tests will be ordered by the consultants or the clinicians as necessary. Because most of these cells are abnormal cells, they are quite fragile, and in a course of film preparation, they break down and form a smeared or smudged cells on the blood film. So one of the observations that will be seen as typical CLL is smudged cells on the film, very rampant in different high power films. The immunophenotyping test will also come up with positive for CD5, CD19, CD20, CD23, and also we will have the kappa light chain of the immunoglobulins or the lambda light chain of the immunoglobulins, and it is mostly either one of them, and giving a clue and confirmation about CLL. Looking at these symptoms, it is something interesting to know, and we should pay attention to these symptoms. If you need further guidance as to how this cause of disease needs to be treated, there are more treatment guidelines in Cancer Research UK, and also you contact your clinicians and they will know the appropriate measures to take in helping you. This is a platform for education, easier learning, making complex things so simple. If you love this kind of education, click the like button, subscribe to this channel, follow me on YouTube, TikTok, Facebook. Let's have a chat together. Have a nice day.